Hello, everybody. In this video lecture, we will cover chapter 27, male reproductive system, and of course, physiology. That's a physiology lecture. So let's go ahead and begin. So talking about physiology, let's go ahead and start with erection. Erection is enlargement and stiffening of penis. During normal activities, arterioles are normally constricted, but sexual excitement causes central nervous system activation of parasympathetic neurons. So during sexual excitement, parasympathetic neurons release chemical called nitric oxide. And nitric oxide causes relaxation of local vascular smooth muscles. So arterioles are normally constricted, right? But nitric oxide causes a local vascular smooth muscle relaxation. Now, when smooth muscles relax, right, then arterioles dilate. I just want to remind you that arterioles, they have the smooth muscles in their walls. Now, in the penis, we have several parts of the penis. It's called corpora. Corpus is body. So we have corpora cavernosa. This is a paired structure inside a penis. And corpora cavernosa expands and retards venous drainage, leading to engorgement of erectile tissue with blood and enlargement and stiffening of penis. Right, so local arterioles dilate, right? Corpora cavernosa fills with blood, right? And this blood is retained within penis, and this causes penis to enlarge. And this has happened because of activation of the parasympathetic nervous system. Why would these parasympathetic neurons get activated? It can be because of sexual stimuli, such as touch, mechanical stimulation of penis, erotic signals, sounds, smells, right? So that's external stimulation of parasympathetic system that causes, again, relaxation of smooth muscles, dilation of local arterioles, and retardation of the venous drainage within penis, specifically structure that is called corpora cavernosa, and penis get enlarged with blood that cause it to be erect and stiff, right? That makes sense. Now, so that's the first part of the physiological process, erection, and then we have ejaculation. Ejaculation is propulsion of semen from male duct system, and this is sympathetic spinal reflex. So erection is parasympathetic response. Ejaculation is sympathetic response. So actually the normal functioning of male reproductive system is a classic example of cooperation between sympathetic and parasympathetic autonomic nervous systems. Okay, so during ejaculation, bladder sphincter muscles constrict, preventing expulsion of urine, because during ejaculation, sperm and urine never mix. That's why the sphincters, constricted sphincters prevent this mixture of urine with the semen. Now, ducts and accessory glands of the male reproductive system contract and empty their contents. Bulbous spongianosis muscles undergo a rapid series of contraction that cause expulsion of semen. Ejaculatory event is called climax or orgasm, right? So, So that's kind of like a first very brief explanation of erection and ejaculation. And of course, you guys need to look at structure of the penis, right, to understand better this physiology process. Either if you maybe already took anatomy class right before, and now this is a human physiology, so we're talking specifically about erection and ejaculation, or if you in anatomy and physiology class, then you will you can see structure of penis in your lab lecture. Now, homeostatic imbalance, it's erectile dysfunction, right? So that's one of these imbalances. And erectile dysfunction, can happen because of parasympathetic neurons of penis release too little nitric oxide. And we know that nitric oxide is needed for smooth muscle relaxation, right, and dilation of arterioles. So possible causes of this little release of nitric oxide can be alcohol, drugs, hormones, problems with cardiovascular system like blood vessels or nervous system, incompetent venous valves that fail to retain blood in the penis. And those drugs that well known like Viagra, they potentiate existing nitric oxide effect. Okay, so that was very briefly erection and ejaculation. So what you guys need to remember over here, that erection is a sympathetic, parasympathetic reflex, and ejaculation is sympathetic reflex. There is some mnemonic to remember it because during this sexual excitement and intercourse in the male body response, we need erection and ejaculation, and we call it like point and shoot. So point, erection, so parasympathetic, start with P, point, parasympathetic, shoot, ejaculation, start with S, so that's a sympathetic reflex. Okay, and another part of this physiology lecture that is really pretty brief is spermatogenesis. Spermatogenesis is production of sperm or spermatozoa in seminiferous tubules. So seminiferous tubules are located within testes. So spermatozoa are gametes, and they have a haploid chromosomal number, abbreviation is N, and for human, our haploid number is 23 chromosomes. So it's only one member of homologous pair. So if you guys remember, diploid cells or somatic cells, they have 23 pairs of chromosomes or 46 chromosomes. So 23 pairs that we all inherited from our parents, from 23 from our mother, 23 from our father, right? That's why as diploid organisms, we have 23 pairs of chromosomes. Now, when we produce gametes, right, sperm or ova, then sperm and ova, they have only one member of homologous chromosome. Now, spermatogenesis occurs in seminiferous tubules of testes that we already mentioned. And this is when spermatogenic cells, and those are stem cells, give rise to sperm. So we go from stem cells, spermatogenic cells, to properly functioning sperm cells, right? That's spermatogenesis. And it can be broken into three phases. Mitosis, of these stem cells, and they called spermatogonia for male reproductive system. So mitosis or spermatogonia that forms two spermatocytes. Next phase is meiosis. And meiosis, when these spermatocytes form secondary spermatocytes and then spermatids. And the last stage is spermiogenesis. This is spermatogenesis, so it's production of sperm. This is, this it's kind of like a bigger process that includes specifically spermiogenesis or spermiogenesis, that when spermatids become sperm, right? It makes sense. It also, we will have a diagram in a minute. So mitosis, you should remember that during mitosis, two diploid daughter cells are formed, right? During meiosis, it's a reduction division. Meiosis forms haploid cells. So we reduce ploidy by half. And then, and spermatids are formed, but spermatids need to become functional sperm, and this has happened during spermiogenesis. Okay, so over here you can see this diagram. So the whole process is spermatogenesis, and the first part is mitosis, meiosis, and spermiogenesis. Not spermata, but spermiogenesis. Right, so that's our three processes. Okay, so the first part is mitosis. So mitosis of spermatogonia forming spermatocytes. So spermatogenesis begins at puberty, begins with spermatogonia or stem cells. Each mitotic division yields one type of A daughter cells and one type of B daughter cells. And A cells, they maintain germ cells lying in the basal lamina. So they stay at basal lamina. I'll show you where it is in seminiferous tubules. So they became new germ cells, new stem cells, right? And type B cells, they become primary spermatocytes, right? So this diagram show us the, all three actually processes, but we can pay attention to this first process that is mitosis. Yes, mitosis. Now, what is this? This is part of the seminiferous tubule. So here's the name. Seminiferous tubule is the tubule inside the testis. Right, so if you imagine like this would be a tube, right? So here's, this doesn't want to work. Okay, so here's a tubule. I don't know, my pen doesn't work for some reason, and very hard for me. Oops, I don't know. Okay, I'm sorry for these drawings, but if this is a tube, right? So the tube is made of cells, right? So here's the cells. One cell. Oh my goodness. Another cell. Yeah, okay, let me just remove this. Yeah, this doesn't really work very good. I'm sorry. Anyway, so again, do some research and see the structure of the structure of testes. And testes are pretty much made of these tubules. They are convoluted, highly convoluted tubule, and wall of the tubule are made of big cells shown here in yellow. And this is called a sustentoside, right? So this, the whole yellow part over here is sustentoside, and this is the nucleus of sustentosides. Another name, maybe you heard about it, Sertoli cells. So these yellow cells are cells that make a wall of those tubules, right? And here's the basal lamina. Now, other cells that are present, so here's the lumen. So here's the lumen of the seminiferous tubule, And from here, sperm will move to epididymis, vastus deferens, right, and ejaculatory tube, and then finally into the urethra, right inside the penis, right, and so on. So that's the lumen. This is where spermatozoa is released, right, and then it's moving to different tubular system of male reproductive system to the exterior, and spermatozoa is ejaculated with the sperm during the intercourse, right? So anyway, so those are sustentosides, and this cell over here, that's a stem cell. For male reproductive system, it's called spermatogonium. Now this spermatogonium divides by mitosis. So this is diploid cells, so it's 2N. So it divides and forms two daughter cells, right? Like what happened during regular mitosis. Now these cells are also diploid, so 2N, 2N. One is called A daughter cell, and it's remain over here at this basal lamina. And these B daughter cells start moving right towards the lumen, right, towards this luminal compartment. And this cell then become primary spermatocytes, right? So this cell, so A cell stays a stem cell. B cells becomes primary spermatocyte. Now this primary spermatocyte then will divide, but not by mitosis, but by meiosis, and form two daughter cells, now haploid, so it's 1N. And these cells will call secondary spermatocytes, right? So here is just a mitosis that tell us that spermatogonia divides, forming A and B daughter cells. A cells maintains germ cell or stem cells, and B cells move towards lumen and develop into primary spermatocytes, right? So now that's our primary spermatocyte, and this is the first part of spermatogenesis, right? So that's our first part of spermatogenesis, mitosis or spermatogonia, right? So now we are in the second stage. So the second stage is meiosis. When we have spermatocytes, and spermatocytes will go through meiosis 1 and meiosis 2. So during meiosis 1, primary spermatocytes that is diploid forms two secondary spermatocytes. So from primary spermatocytes to two secondary spermatocytes that already haploid. And meiosis 2 forms 4, right? Each one. So we form 2 over here. Each of them divide one more time. So we form 4 spermatids, right? That also haploid. Spermatids are small, non-motile cells found close to lumen of tubule, right? So here's our diagram again. So here's our primary spermatocytes divide through meiosis. So we have 2 divisions, meiosis 1, meiosis 2. 2 cells, both haploid, they called secondary spermatocytes, then they divide one more time, forming 4 total of 4 cells, each haploid, and it's called early spermatids. And they small, they don't have tail yet, right? They have extra cytoplasm. Mitochondria is not organized in the mid region, right? So those are just early cells that will develop further into functional sperm or spermatozoa. So sperm is the same as spermatozoa, right? So that was stage 2, that is meiosis. And then stage 3, spermiogenesis, when these spermatids become sperm. So spermatids contain correct haploid chromosome number needed for fertilization, 1N, right, 23 chromosomes, but they are non-motile. And spermiogenesis is a streamlining process where spermatids elongates, loses excess cytoplasm, and forms a tail, and they become spermatozoon or sperm. And when we look at region of the sperm, we have head, midpiece, and tail. So head is genetic region that include nucleus and acrosome. Acrosome contain hydrolytic enzymes that enable sperm to penetrate egg. Midpiece is metabolic region containing mitochondria that produce ATP to move tail, and tail is locomotor region that includes flagellum. Right, so over here we can see this is our spermiogenesis, formation of sperm. So we have spermatid, right? So that's our spermatid, and during spermiogenesis, we end up with sperm, right? So you see a big difference in the in appearance of these cells, how they look. Right now, so what happened over here that these acrosomal vesicles develop, right? So here's the Golgi apparatus. This is nucleus. So we have a lot of synthesis going on over here, right? Because we need to synthesize those enzymes that will allow the sperm to penetrate the egg, right? So, so all these proteins, enzymes are synthesized. They are accumulated in the organelles that form this acrosome. So those are acrosomal vesicle. Now also we have rearrangement of mitochondria. Excess cytoplasm is lost, right? So we don't need cytoplasm. We don't need, you know, any really organelles except of nucleus, acrosome. Mitochondria form this mid-piece region, right? And then where we have the centrioles, right? The tail is formed, right? Made of microtubules, and this tail is called flagellum, right? So that's the sperm that has head region, mid-piece region. Mid-piece region contains mitochondria, and this is locomotion, and tail. Oh, this is for energy, and tail for locomotion. And head is for blueprint, for genetic information. Okay, now that was 3 stages, right, of spermatogenesis, right? So mitosis, meiosis, and spermiogenesis. Right, over here we can see spermatogenesis, mitosis, meiosis, spermiogenesis. Okay, now role of sustentosides. So sustentosides, again, I want to remind you, are those large cells shown here in yellow color, and this blue region is nucleus of this sustentoside. Okay, so role of sustentosides. They are large supporting cells, also called Sertoli cells. They provide nutrients and signals to dividing cells, secrete testicular fluid into lumen for sperm transport, phagocytize faulty germ cells and excess cytoplasm, produce chemical mediators to regulate spermatogenesis. So you can see, you know, lots of function for these sustentosides. Now, if you look over here, this part, so you can see that these cells, they actually have tight junctions between them. So they really connected to each other, and those tight junctions form blood-testis barrier. Now, why do we need this blood-testis barrier? Because sperm is not formed until puberty. So it is absent during immune system development. And when your immune system develops, your white blood cells, they selected, right? We have like positive and negative selection, but cells of your immune system selected to attack all foreign proteins and tolerate your own cell markers, right? So your immune system, or cells of your immune system, produce antibodies, complement and other type of defense against everything that is antigen. So foreign antigens are attacked by the cells of your immune system. So because your immune system formed during early development, it's recognized your cells, but sperm are not present at that point. So sperm are actually presumed by your immune system as a foreign cells, right? Like something that doesn't belong to you. It's not non-self, right? So because the sperm is not present during immune system development, it's recognized. It cannot be recognized as self, and it's recognized as non-self, right? That's why we need to keep sperm separated from your blood. Because if sperm somehow gets into blood, right, or those white blood cells can get inside the seminiferous tubules, then they would attack your own sperm, right? So therefore, sperm needs to be kept separated from rest of body to avoid being attacked by immune system. That's why we have this tight junction between Sertoli or Sustentosides to prevent your immune system from attacking this newly formed sperm. Okay, so how long spermatogenesis takes place, right? It takes about, from a little bit more than two to 3 months, or 64 to 72 days if conditions are hospitable. Sperm are unable to swim, but pressure of testicular fluid pushes immotile sperm into epididymis. So when sperm are formed over here inside seminiferous tubules, even they do have this flagellum, they cannot swim yet. But the same cells, Sustentosides, they produce the fluid, and pressure of this fluid gonna push the sperm inside epididymis that is located on superior, I think, lateral part of the testis. And in epididymis, this is where sperm gain motility and fertilizing power. So they mature in the epididymis. So they formed in seminiferous tubules inside testis, then they pushed into epididymis, and that's where they mature. Now, obviously, everything is regulated by your endocrine system, because that's what keeps us in homeostasis. We need regulation, and spermatogenesis need to be regulated as well. Now, the systems that regulate, or I would say the systems that regulate the spermatogenesis is endocrine system, right? You can say nervous system included as well, because hypothalamus is part of nervous and endocrine system, but majorly regulated by endocrine system. But the process that regulated, it's called HPG axis, or hypothalamic pituitary gonadal axis. So you can see from the name, it starts at hypothalamus, then pituitary, anterior pituitary is included, and then gonads, in this example, are testes, right? So production of gametes and sex hormones is regulated by sequence of hormonal events involving hypothalamus, anterior pituitary gland, and testes. And it's referred as HPG axis. And hormones that are involved are GnRH, or gonadotropin releasing hormone, FSH, LH, follicle stimulating hormone, luteinizing hormone, testosterone, and inhibin, right? So, and we will talk about this process in a second. Okay, so hypothalamus releases gonadotropin releasing hormone, GnRH. GnRH binds to anterior pituitary gonadotropic cells, causes them to secrete FSH and LH. Now, this FSH and LH, they are tropic hormone. That means they have target. Their target is another endocrine gland, and you can guess what endocrine gland is their target. testes, right? And ovaries as well. So keep it in mind. We will talk about the same hormones when we cover female reproductive system. So FSH stimulates spermatogenesis indirectly by stimulating sustentosides to release AB protein or androgen binding protein. AB protein keeps concentration of testosterone high near spermatogenic cells, promoting spermatogenesis. Luteinizing hormone binds to interstitial endocrine cells, prodding them to secrete testosterone, and rising testosterone level triggers spermatogenesis. Okay, let's do this because we have, you see, we have 7 steps over here. Let's look at diagram first, and then, we will get back to that list. Okay, so here's your hypothalamus. This is pituitary. Here's anterior pituitary, posterior pituitary, and those are testes, right? Everything else are testes, specifically seminiferous tubules. Now, hypothalamus release gonadotropin releasing hormone. All hormones are released by hypothalamus, if I'm not mistaken, but never say all, right? They might discover something else, or maybe they already did, and I just don't know it, but let's say hormones that released by hypothalamus have in their name, they have word releasing. So gonadotropin releasing hormone, this hormone stimulates gonadotropin cells of anterior pituitary. So they stimulate anterior pituitary. Anterior pituitary release FSH and LH. Now FSH, it activates these sustentosides or Sertoli cells, and Sertoli cells release AB protein. And what this AB protein does, it keeps testosterone level over here high. And when testosterone level in this area high, then it stimulates spermatogenic cells to divide. So it stimulates spermatogenesis. All right. Now, luteinizing hormone activates interstitial endocrine cells. Interstitial means they are inside this tissue of seminiferous tubule. Those are endocrine cells, and they produce testosterone. So we need this testosterone, right, for spermatogenesis, for spermatogonia to divide, and for sperm to develop normally, right? So testosterone is released by interstitial cells. AB proteins maintain high testosterone level, and by that, doing this, it promotes division of the spermatocytes, right? Also, a part of testosterone is secreted into blood, and this has some somatic and psychological effect, such as body size, maintain secondary sex characteristic, and so on, right? But spermatogenesis cannot process normally if we have low testosterone level, right? So a release of testosterone depends on anterior pituitary and hypothalamus, right? So from hypothalamus to anterior pituitary to Sertoli cells, interstitial cells, testosterone level goes up. At the same time, this system needs feedback mechanism. So when testosterone level increases, it inhibits anterior pituitary, right? So that's our feedback mechanism. Too much testosterone, inhibition of anterior pituitary, less FSH, LH, less testosterone, right? Because that's need to be in balance. Also, high count of sperm, right? So you have too high count, too many spermatocytes, then this stimulates Sertoli cells to release inhibin, and inhibin also inhibit anterior pituitary and hypothalamus. So it's another feedback mechanism. So here we have all our 7 processes. So if you go back, right, so that's all of them. So you can read it right? But it's pretty much what I just explained you, right? So these lines, these arrows show us stimulation, and these dotted lines show inhibition. So it's a perfectly balanced process that has shut-off mechanism, right? That has turn-on mechanism, and so on, right? So like the question would be, well, let's say if somebody has low count of sperm, what can we do? So if we increase the testosterone level, we cannot increase this too much, because too much testosterone actually gonna inhibit right spermatogenesis. Okay, so so effects of testosterone. Testosterone synthesized from cholesterol is transformed at some target cells, from spermatogenesis and target all accessory organs, has multiple anabolic effects through body. And remember, anabolic is when you build something, your body builds larger molecules, right? So it's an endergonic process. Deficiency of testosterone leads to atrophy of accessory organs, semen volume declines, erection, ejaculation impaired, and treatment is testosterone replacement, right? So let's say, of course, when we have, problem with erection, ejaculation, we can treat it with the drugs that promote release of nitric oxide, right? But again, I see if the problem are in a low testosterone, testosterone can be given, but too much testosterone, again, it will at the same time decrease the sperm count, because it has this negative feedback loop, right? High testosterone level. Okay. Another effect of testosterone is that it would gives us a secondary male characteristic, like appearance of pubic, axillary, facial hair, enhanced growth of chest hair, deepening of voice, bones grow, increase in density, skeletal muscle increase in size and mass, boost basal metabolic rate, and basis of sex drives or libido in males. Um, Testosterone also masculinizes embryonic brain, continues to exert effect well into adulthood, and also we know that endocrine glands, I'm sorry, yeah, endocrine glands that are adrenal glands produce androgens, and testosterone is an androgen, but adrenal gland produces it in a small amount that is not sufficient to maintain normal testosterone mediated functions. I think that's it. So as I said, it's not very long chapter. Please make sure that you look at structure of, if you forgot, please review the structure of penis, anatomy of penis, anatomy of testes, and then you will understand this physiology part better. Okay, well, thank you for watching, and I hope it was helpful.